My name is Francesca, and I'm a structural bioinformatician at Accenture, where I've been working for almost 3 years now. I'm very excited to discuss with you the work my team and I have been doing around automating bioinformatic pipelines to enable drug discovery at scale. Accenture is an AI-driven precision medicine company committed to discovering, designing, and developing the best possible drugs in the fastest and most effective manner. It was the first company to progress AI-designed small molecules into the clinical setting by focusing on encoding and automating all steps of drug discovery. By pioneering a new approach to creating medicines, we believe that the best ideas of science rapidly become the best medicine for patients. Our platform is a densely connected design loop, and I've personally been working on the target and design sides of this diagram, using bioinformatics methodologies to support both target identification and small molecule design. Our platform teams are modular and cross-functional to enable the development of autonomous subsystems that benefit from domain expertise in AI and computational design. In this way, we do not seek to replace our designers, but rather learn from their intuition to develop better and better heuristics and algorithms. Several tasks that fall within the structural bioinformatics remit can be automated, and we have done that by treating 3D data with the same attention that has traditionally been given to 2D ligand data. This has allowed us to gain insights at several points of our design process, from robustly and accurately preparing public and internal structures to automatically generating design ideas. One of the key technologies we use to exploit structural data is Fragment Hotspot maps, a statistically driven approach that is built on a detailed analysis of more than 1 million small molecule and protein ligand structural datasets. It maps the binding pocket to describe the size, shape, and electronic composition of molecules likely to bind. These maps can be combined and subtracted from each other to allow us to understand areas of commonality between targets or areas of difference that can be used to drive selectivity. Methodologies like hotspot maps are combined in a modular fashion within several different automated pipelines, of which I will discuss 2 examples. One pipeline for target identification and assessment, and one for the automated generation of kinase inhibitors. The target identification problem deals with assessing how well suited a new protein target would be to our design methodology. Part of that is to use hotspot maps to understand how druggable a given binding site would be. Hotspot maps quantify the druggability of a protein structure at any given point and can be combined to represent the attractiveness of the pocket as a whole for drug-like molecules. Additionally, these tractability maps can find distinct binding pockets on a protein structure. To produce a druggability assessment of the target as a whole, we cluster the residue composition of each binding pocket on each protein structure to retrieve global binding pockets that can easily be indexed and retrieved by our workflows. My colleague Jake is presenting poster 721 and will be able to describe this clustering method in detail to those of you that might be interested. Overall, the workflow is as illustrated in this diagram. Firstly, available structural data is collated. Then, a frame of reference is selected based on quality metrics, and all structures are then prepared and aligned to this frame of reference. Then, hotspot maps are calculated for each structure and transformed into tractability scores. Finally, global binding pockets are identified for a given target. This is an example plot of the output of our tractability analysis. Here, the tractability score distribution of an example kinase target are plotted in orange to allow us to compare the target to the scores of known druggable and not druggable proteins. Clustering binding pockets across a target allows us to be sure we are looking at the scores of the main kinase binding pocket, while still being able to identify structures that score higher or lower, due for example to conformational movement. Once global binding pockets have been identified, we can expand our analysis by incorporating data relative to the residues that make up a given pocket. For example, we can compare hotspot scores with mutation data to identify those positions that are both highly druggable and likely to be robust to future mutation, paving the way for design strategies that favor variation-resistant interactions. The second automated pipeline I'd like to discuss is the one we developed to generate de novo designs for kinase inhibitors. The aim of this project was to establish whether it would be possible to go from a structure all the way to a first series of synthesizable designs in a fully automated manner. we have shown this to be possible across multiple kinase structures with a generalized, cost-effective automated pipeline that has the potential to be scaled to high volumes of targets. We started this work around the time the AlphaFold2 database was published, which provided structural models across the whole proteome. We ran the target assessment pipeline, which I've described earlier, across the AlphaFold models for the whole kinome, and obtained a list of druggable structures for which no experimental structure has been solved. To help us understand how model quality correlates with the quality of output compounds, we selected a subset of kinase targets that varied both in model quality and in sequence similarity to the closest available experimental structure. In this talk, I will discuss results of running the pipeline on two targets that are at opposite ends, DYRK1B, which is very similar to an available structure, DYRK1A, and PKD1, which is very dissimilar to the next closest kinase. The first step of this pipeline is to gather a set of suitable ligands that will be fragmented to yield seed fragments for our generative algorithm. For this proof of concept, given we were focusing on kinase structures, we used the CLIFS ligand set, which contains known kinase inhibitors, but no compounds associated with our specific targets. We have also been working on alternative strategies to generate suitable ligand sets, even for non-kinase targets. One of these strategies pulls in ligands from homologous structures, and I recommend you drop by poster 721 if you'd like to hear more about it from Jake. Each molecule in the input ligand set is then trimmed down to a key pharmacophore using the hotspot maps. Indeed, we know that the part of a compound that binds to the hotspot will contribute to the binding affinity much more than the rest of the molecule. Therefore, we can trim the molecule back to the part located in the hotspot, knowing that the binding mode will be conserved. Similarly, we expect compounds grown out of that same fragment to bind to the protein target in the same pose. While we trim every ligand in the initial set, we select only 50 fragments to go through to the next step, maximizing their chemical diversity and hotspot complementarity. The selected fragments can then be grown again using our own generative algorithm, Gambit. Gambit efficiently searches chemical space for molecules that fulfill a set of defined objectives. Additionally, Gambit only grows compounds from fragments using chemically allowed steps, in other words, synthetically possible substitutions. This process is repeated over and over again, iterating over the best scoring compounds. In this pipeline, several parallel Gambit runs are kicked off, with a reward function that maximizes synthetic accessibility, drug likeness, potency, similarity to the original fragment position, and hotspot map complementarity. Each run is expected to output 250,000 compounds, but many millions of molecules are sampled to efficiently explore the chemical space around the fragments. These compounds are then scored and filtered in parallel to whittle them down to our final compound selection. The filtering part of the pipeline aims to select compounds with desirable properties, such as molecular weight and minimal conformational strain. Additionally, a subset of compounds is selected using coverage score to maximize their chemical diversity, and then that subset is filtered based on binding site interactions and synthetic accessibility. In this way, we yield 50 compounds per target, and here you can see the 50 compounds we generated for DYRK1B. All of these are novel compounds, and we commissioned the 9 easiest to synthesize to be made and assayed. Similarly, these are the 50 compounds generated for PKD1. Of the 9 compounds synthesized for DYRK1B, 4 were found to successfully inhibit our target, with IC50s ranging from 42 nanomolar up to around 1 micromolar. Additionally, we can see here that they were assayed against a whole series of kinase targets, and that they are selective to DYRK1B. Similarly, 4 out of 9 compounds were found to successfully inhibit PKD1, mostly in the 1 micromolar range, but one as low as 73 nanomolar. We can see from the data presented on this slide that they do not show the same inhibitory behavior with the other targets they were assayed against. If we look at the ADME data for our DYRK1B hits, we can see that there are some fairly attractive profiles, such as compounds 0715, which could be considered to be a reasonable starting point for further design iterations. The same can be said for a PKD1 hit. There are clearly properties, such as solubility, that need to be addressed and iterated upon, but this is expected of hit-finding processes. Typically, initial design ideas would be subjected to 2 or 3 exploratory design cycles to help develop a base understanding of on-target biology and ADME profiles. The autonomous pipeline I've just described will be extremely useful at the start of a project, allowing designers to be presented immediately with a fast design series. Overall, I've presented 2 end-to-end bioinformatics workflows that are already in use in target evaluation and hit ID processes at Accenture. On the target identification side, multi-scale data aggregation allows users to have both a big picture overview of a given target and detailed information about relevant structural outliers. On the ligand generation side, we have demonstrated the usability of lower confidence AlphaFold models, with nanomolar hits designed for PKD1, opening the possibility for kinome-wide design. Furthermore, the same automated workflow could be applied to non-kinase targets by leveraging PDB-wide hotspot maps and custom input ligand datasets. Above all, the autonomous and modular nature of all our workflows enables constant improvements and iterations, allowing the pipelines to keep up with the cutting-edge science produced by our research teams. Speaking of which, I'd like to thank my colleagues for their contribution to the work I've presented here today, and I'd like to thank you all for listening.